Now, I wanted to discuss about AFLP, that is Amplified Fragment Length Polymorphism. This is a PCR based marker technique. Just before that, I just want to go back to RIPD and RFLP because both of them having some sort of setbacks with them. Because in the case of rapid, the results are quite complex, so sometimes it may be hard to interpret. And the second issue with rapid is that the result of rapid, in most cases, it cannot reproduce. In the second case, that is in the case of RFLP, here we require lot much sample DNA to work with. That's why we are much more interested in AFLP. Just before going into the basic principle of AFLP, I just want to tell you about linkers and adapters. What are linkers? These linkers are a double-stranded short oligonucleotide DNA having arrestation sites within it. They are double-stranded, chemically synthesized DNA with one or more arrestation sites within it. So, how these linkers can help to solve the above mentioned issues? This is the gene of interest, and this is the vector in which you wanted to introduce this particular piece of DNA. But the problem is that this vector having limited choice of restriction sites within it. Suppose it's having only two restriction sites within it, then for example echo R1 and BAMH1, then you have to excise the DNA or gene of interest with either of these restriction enzymes. But here also lies another problem, that is if you are using echo1 here to excise the DNA, then sometimes this gene of interest may have echo1 resident sites within it. Then it poses another problem. If you uses the echo1 resident enzyme, then that cuts the DNA of interest within it. So that severs your gene of interest into several pieces. So that's not a good choice at all. Then the another choice is that BAMH1. But here lies another problem, that if your gene of interest is not overlapped with BAMH1 site, then what will you do? Here you can depend on linker DNA. I have told you just a while ago that these linker DNA are short double-stranded DNA having restriction sites within it. Okay. The first thing you have to do is that you have to excise the DNA with some other combinations of restriction enzymes. Keep in mind that that doesn't harm your gene of interest. Suppose this had been excised with SAL1 and HIN3. Okay, then you might have SAL1 here and HIN3 may be over here. Okay, but you cannot introduce this DNA into your vector. So to solve this issue, you have to ligate linker DNA on both ends of this gene of interest. So this is gene of interest and this is the ligated linker DNA. Okay, linker DNA, gene of interest, linker DNA. This linker DNA also having BAMH1 resident site also within it. Then you have to treat it with the BAMH1 restriction enzyme. So that BAMH1 will cut on both the of the linker. So here you will get a sticky end, and here also you will get a sticky end. That is, resident sites are opened. Then you can introduce it into the vector that is opened with the Babbage one. Now, what are adapters? These adapters are also having somewhat a similar structure with that of a linker, because this is also a chemically synthesized double-stranded oligonucleotide that having a short single-stranded overhang. This overhang can be developed as a Russian site. Then, here in the case of a linker, you have to treat the linker with the rustration enzymes to make it sticky. But here, it's already sticky in nature on its own end. This end that can be used to ligate to the DNA, and this is already cut open one. So there is no need to treat it with the rustration enzyme further. Coming back to the AFLP, this is the genome or DNA which you have obtained from some of the sources or the sample DNA. The first thing you have to treat it with a rustration enzyme or combinations of rustration enzymes. Then that rustration enzymes will sever your DNA into several fragments. Suppose these reactions are carried out in a tube, a kind of tube. Okay, that means you first of all you have to take this genome or DNA into an appendage tube, and then you have to add rustration enzyme into it. Then you have to incubate it. Then what happens is that that rustration enzyme will cut the DNA into several fragments. Okay, this is happening in a tube. Next thing, you have to add adapters onto it. Okay, adapters. After that, you have to add ligase enzyme. Then what is happening is that this ligase enzyme ligates this adapter into each and every fragment that had been produced during a restriction. And this is actually depicted here in this picture. That is, here is the genome. Exactly the similar thing, the same thing. This had been digested with restriction enzymes. Don't think about this, okay? Then you will get so many fragments. This is ornament them. And next thing, you had added adapters. Here in this picture, they had used two adapters, two types of adapters. They are echo-run adapters and MSC1 adapters. Okay. And the next step is that you had added a ligase enzyme onto the reaction tube. Then what is happening is that this is your DNA. This is one of the DNA fragment, and this is an adapter, and this is also an adapter. After adding this ligase enzyme, these adapter will join over here, and these adapter will join over here. Okay, that means these fragments will get ligated. Okay, clear. So now the entire this adapter plus these fragment plus this adapter. So that is shown over here. Okay, this adapter, DNA fragment adapter. Okay. Normally during the PCR process, you cannot amplify this particular DNA fragment, this particular DNA fragment. You know why? Because this this particular fragment is coming from somewhere in the genome. The sequence of it might not be known, so you cannot synthesize or prepare a primer against it. Okay, but you had already added adapters on both of its ends. Okay, this is echo-run adapter and this is MRC1 adapter. These sequences of these adapters may be known. Okay, the sequence of this thing and this thing is already known. So you can definitely prepare primers against this Ecoron adapter, against this MRC1 adapter. So what is happening is that this primer will bind over here and this primer will bind over here, and during the PCR reaction, this primer may be extended and this also will get extended. That means this particular fragment get amplified. Okay, okay, this is amplified product. Now during the normal RFLP reactions, what is happening is that your genome will be fragmented into so many pieces by means of precision enzymes, and that fragmented sample will be loaded onto gel. Then there in the gel you will get a pattern. But what is happening in the AFLP is that you will have to digest it with the rust enzyme, exactly similar to that of RFLP, and then thereafter each fragment will be amplified with the help of a PCR. So here in the gel you will get amplified fragments. Okay, what is the difference between the RFLP and the AFLP is that here in the AFLP each of the fragment will get amplified. So you will get a better resolution in AFLP. Okay.